So good morning, everyone. So my name is Hoi Man Kevin. My supervisor is Dr. Yuan Huang, and I'm from the University of Hong Kong. Today, I'm going to present you my manuscript, Interpretable Variational Encoding of Genotypes Identifies ISMB clonality and lineages in single cells geometrically. So first of all, I want to go through some background knowledge, the motivation of my project, which is the mitochondrial lineage tracing. So almost all eukaryotic cells have mitochondria inside, and each mitochondria contains several copies of DNA. So as a result, a single eukaryotic cell contains many copies of mitochondrial DNA inside. And although each mitochondrial DNA only has 16,000 base pairs, the mutation rate is high compared to nuclear DNA. And given all these advantages of them over the nuclear genome, we find that it's maybe a good candidate for lineage tracing in natural cells, where you don't need to edit it by CRISPR or some other gene editing, but then you still may capture the lineage information. And there is not much method really about mitochondrial phylogeny published. So we also and our collaborators are very interested in this kind of mitochondrial lineage tracing. So we developed a computation method for that. It's called SNP manifold. So it's a neural network model which encodes discrete single cell genotypes in the continuous space. And this is the layout of our model. You can just focus on the top right corner, which is the backbone model. It's a neural network using binomial variational autoencoder. Autoencoder is a type of popular neural network architecture that is widely adopted and solve many problems. So we want to apply it to single cell genotypes. But then we need to replace the cost function because the ordinary cost function is Gaussian, which is not suited for the genotype, which is LA, LB count. Those type of genotypes, they follow a binomial distribution instead. So we change the cost function, and we also engineer the architecture a bit and then finally we realized that by reducing the layer really a lot, because we only have one hidden layer here, unlike ordinary neural network, they have many hidden layers. But then we find out that by having only one hidden layer and some other transformation tuned, we find out that this latent space, which is a 2D matrix of cell and latent vectors. So we can learn the latent vectors for each cell, and then we find that these latent vectors is a good representation of the single cell genotypes because they can extract a pattern more clearly than the raw signals. So based on this latent space, we can perform clustering and then the parent selection and also the phylogeny. So this is a results plot. So we first try it on a single cell data set from 4 discrete donors. So 4 donors, so there are 4 discrete genotypes. We first want to use them to feature extract by using the probabilistic PCA and UMAP, which is on top, and then there are 2 baseline methods, and we find that although they can separate the donors a bit, but they are not really extracting the patterns. And then our SNP manifold is down below, and you can see that's now the 4 the cell separates into 4 clusters really clearly in the latent space. So we believe that our latent space has the power to really manifesting the genotype pattern. And then this is the mitochondrial data set, which is the core mission of my project. So this is a mitochondrial data set of 2 cell line mix, so 2 cell lines from really distinct origin, and mixing them together, and then to see what are the phylogeny inference based on them. So because there are 2 cell lines, so they are of distinct origins, and then also inside each cell line there are somatic mutations, and we want to infer the phylogenetic tree inside our latent space. And we find out that there is a clear cut between the 2 cell lines, which is cut by the red line. And then on the left of the tree, it's 1 cell line, and on the right of the tree, it's another cell line. So we believe that a phylogenetic tree constructed in the latent space are still of considerable confidence, and we may kind of believe in it. And then this is another data set, which is 1 single cancer cell line, so all mutations are somatic. And we realized that there are 2 parts of the mitochondrial genotype. So you can see that there is 1 big cluster of cell down there, and then one that is up here that is just significantly separate from the rest of the cells. And then we also find out that this separate mitochondrial DNA clone also has distinct CMV in the nuclear genome. So this is quite interesting because distinct CMV, of course, in the case that they come from distinct origin. And then our analysis on the mitochondrial DNA also proves that they are also coming from, they also have distinct set of mitochondrial mutations. So this may kind of prove the tracing power of mitochondria. And then the phylogenetic tree constructed by our method can kind of identify a possible origin of these CNV cells to the closest normal cells. So finally, I also want to highlight this data set. So this is a data set with HSPCs, the blood stem cells, and also a PBMCs, which is a peripheral blood cells collected 3 months later. So because there is a 3-month interval, so there will be sufficient time for the HSPCs to differentiate into a PBMCs. So, and we also expect a direct lineage relationship between them. So this is also a mitochondrial data set. And then the phylogenetic tree constructed by our method shows that the HSPCs are in the middle and the PBMCs are on the periphery, on the peripheral terminal nodes. So you can see the color of the tree. So basically deeper color indicates more stem cells, and then the lighter color indicates more of the peripheral blood cells. And we realized that our method successfully identified the common ancestor, which they come from a small group of stem cells and then but differentiate into different lineages of PBMCs. And then finally, I want to highlight that we go back to nuclear DNA. So this is nuclear DNA dataset, and we realize that our method also outperforms some other existing methods by being able to detect more rare mutations with cell numbers around 1% of the total. So summary. So basically, SNP manifold can effectively identify SNV patterns in single-cell mitochondrial DNA. So it may be useful in lineage tracing, and it's more sensitive to rare SNVs with low cell number. So it may be also useful in single-cell cancer phylogenetics. And our manuscript are available on bioRxiv and also the code on GitHub. And thank you for Dr. Huang for my supervision of my project.
What is the resolution? I mean, how low could you go? How small subclones could you still detect with your method? Did you check this?
For this one?
Yes. Yeah.
Yeah. So basically, it is around 9,000 cells. But then, for the smallest donors we mix, it's around only 60, 70, 80 cells. So it's dividing it, it's only around 1% of the total. But then we are still able to detect it, while other methods that are designed, that are published before, they will kind of make some errors by combining the rare clones or misclustering.
Do you see any possibility to increase the resolution to find even smaller sets of cells?
I think that depends on the phylogenetic distance and also the cell. Because when the cell number is low, then you need a really larger phylogenetic distance difference to separate them accordingly. So it depends on the distance.
Thank you.
Hi, really amazing talk. I have 2 quick questions. First one is, so here you're running it on the nuclear DNA. So your method is not specific to mitochondrial DNA. Is that true?
My method is general to genotypes because it uses a binomial likelihood and all kinds of allele. It's just that's the nuclear DNA that the number of depth, the sequencing depth is 2, and the mitochondrial DNA, the sequencing depth is more. But then they all follow the same likelihood function, so it can be used by my model.
Cool. So it's just motivated by the mitochondrial DNA to some extent.
The original motivation is for the mitochondrial DNA. So that's why we adopt the binomial likelihood, but then we also realize that it also outperforms existing method in the nuclear DNA.
Cool. Second question is about what's the motivation behind I guess projecting to a latent space instead of using just the SNP themselves?
I think the motivation is really to make the pattern more obvious. It's of course, using the raw space, you can see that like PCA and UMAP, there are 2 kinds of visualization, and then the data points are quite sparse because there are a lot of missing signals and then a lot of low depth sequencing regions. But then our method can kind of account for these errors, and then to make the pattern really obvious and separate into 4 distinct clusters.
Thank you.